Hello students, welcome to EPG Pathshala. I am Dr. Vibha Dhawan from TERI. Today we are going to talk on module Medium Constituents and its Preparation for Plant Tissue Culture. It's under the paper Plant Biotechnology and Crop Improvement. What are you going to learn in this module is that what are the major constituents of tissue culture media? How do you decide what is required by the growing culture and how media is prepared? Now, why all this learning is very important for you is that we are growing very small tissue masses, even single cells, with no maternal influence. So there is hardly any explant tissue and you are growing those cell aggregates or single cells in culture. And they are not going to say anything of their own. It is you who have to decide based on the media. This is what is favoring growth and this is what is leading to their death. So therefore, you have to be very choosy. Then you look into what are the media constituents. They are so large. What are the concentrations that they require? Because most of the time there is no correlation with what the plant likes when it's growing outside, whether it's a heavy feeder or a low feeder versus what it will be requiring in culture. Then there are so many constituents and there are lots and lots of considerations. Is that chemical readily available? Number one. What is the cost of that chemical? How do I dispose that particular constituent? Is it harmful to the environment at any stage? So you have to take all those things into consideration. And lastly, but not the least, when you prepare media, then also you have to be careful because ultimately whatever you are adding should remain available to the plant. So there are certain chemicals which are thermolabile, which get degraded at high temperature, and therefore there are different processes for sterilizing those particular chemicals. When we look into the media constituents, we classify them as macro or major elements, microelements, growth regulators, and vitamins. So these becomes the major constituents of any media. Now major elements, now it's something like what is also required by the growing plant, and their concentrations are more than 0.5 micromole per liter. So that is what we classify as major elements or macro elements. Under this category comes nitrogen, phosphorus, sulfur, calcium, potassium, and magnesium. Some of these, like when we look at nitrogen, then they are either added as nitrate or as ammonia. Phosphorus, again, phosphorus, sulfur, that again is in phosphate and sulfate conjugates. For calcium, potassium, magnesium, they can be taken as standalone elements, and then you can add various combinations like magnesium can be with sulfate, calcium can be with phosphate, potassium hydroxide, and so on. So you can add various conjugates of these particular chemicals. Micro, as the name suggests, are those elements which are required in very small quantities. In this category, we have chemicals such as iron, manganese, copper, zinc, boron, molybdenum, and cobalt. I'd like to mention that the initial media which were prepared were very, very simple, and people or scientists succeeded at this stage because they were growing cultures of species which today we know are highly responsive. White, and way back in 1943, and thereafter Gottfried in 1939, they were the first to develop a nutrient solution media on which all later formulations are based on. I'll also like to mention that it's over a century of research on plant tissue culture media, but even today we can't say that there is any media which can be called universal. So therefore, all the time you have to test what will grow on what particular species. Apart from major and minor salts, it was discovered that iron plays a very important role in the growth. It was Heller way back in 1953 who made a detailed study of inorganic nutrients in tissue culture media, and he gave special emphasis to iron and nitrogen. White was the first to incorporate in 1943 ferrous sulfate in his media, and thereafter Street and Henshaw introduced ferric chloride, and then it was found that these compounds are not very stable, and as the media pH changed, they became non-available to the growing tissue and the deficiency symptoms start appearing. And thereafter, the further development was more towards developing iron chelate and ferric EDTA, a combination of ferrous sulfate and sodium EDTA is the most commonly used source in tissue culture media. In fact, in MS, Murashige and Skoog's media, FeEDTA is used. And this iron source, you make the stock solution of iron and keep it in dark bottles so as it is prevented from any degradation, photooxidation, and so on. Well, you all must have noticed that when we are taught about food, one of the important constituents we talk about is vitamins. You should eat green vegetables, you should drink milk, this contains vitamin A, this contains vitamin B. So there are various sources which we mentioned that should be incorporated into your diet. And still we do talk about deficiency symptoms in animals, but in case of plants, normally we don't talk about vitamins. So the plants have unique ability to synthesize vitamins, which animals and human beings lack. For in most cases, of course, vitamin D is synthesized in humans also from sunlight, but in case of plants, we don't give any artificial vitamins. But what happens is that once you excise or you have very small cell mass or tissue growing or small organ growing, then they are unable to synthesize their own vitamins, and you have to understand and incorporate these vitamins into the tissue culture media. The most commonly used vitamins are B1, B3, B6, and B complex. We meet them through various sources, like B1 is through thiamine, B3 is through niacin, B6 pyridoxine, B complex myoinositol. Then vitamin C, that is largely to prevent oxidation of phenols. Vitamin D, it's more regulatory. Vitamin E, which is again an antioxidant. Riboflavin, which improves shoot growth, prevent callus formation, and leads to direct root formation. But these are some of the examples, and the list is very long and it varies from species to species. However, many times we are unable to identify through vitamin addition and so on what the plant is requiring, and we resort to adding complex substances such as casein hydrolysate, coconut milk, maize extract, tomato extract, yeast extract, and so on. However, we are not very comfortable because you will realize that coconut milk, you really don't know what is the stage of coconut when you are collecting that. So each batch of coconut milk might vary. So therefore, to get uniform response, you don't want to add things like coconut milk, maize extract, tomato extract, and so on. However, I must say that for many species, we are still unable to identify, and we have no alternative but to keep on using these substances. Further, coconut milk really proves to be cheaper compared to many other synthetic chemicals which are available in the market. And there are species which are not very sensitive. Like if you go for orchid tissue culture, that does so well in coconut milk, and there is no influence of the stage of coconut development when you extract coconut milk. and people also develop an eye towards it, how transparent coconut water is, that it should be added or not added to the tissue culture media. All living cells, they require energy. Animals, they don't produce their own energy, and that is why they are dependent on getting food from outside. On the other hand, plants photosynthesize. They prepare their own food from carbon dioxide and water. So it's, and CO2 plus H2O, that gives rise to C6H12O6, that is a carbon molecule or a glucose molecule which is produced. So what happens in case of tissue cultures is that the plant is not, or your cells or tissues, they are not photosynthesizing, or they are photosynthesizing at suboptimal level, and therefore you have to give sugar artificially. Now, what are the sources of sugar? You can get them from wide range of sources such as sucrose, glucose, fructose, maltose, galactose, mannose, lactose, and so on. But we largely use sucrose. It is the best source of sugar, readily available, cheapest of all these sources, and is suitable for most plant species. It's used in the concentration anywhere from 2%, that is for tissue culture, 2, 3, 4%, and it goes as much as 9% when your objective is to induce tuberization in potato. I'll also like to mention that carbon sources, they provide energy as well as they maintain osmotic potential in cultures. If one has to put success of tissue culture to one factor, I'll say it was discovery of Skoog and Miller that the organogenesis, morphogenetic responses are controlled through the interplay of growth regulators. So it's the discovery of growth regulators as well as understanding roles of different regulators in plant systems that has led to various morphogenic responses that we have got in tissue culture. If we want to classify them broadly, the growth regulators can be classified into auxins, cytokinins, gibberellins, and ethylene. Their roles are almost similar to what we get ex agar in nature. We all know that to induce rooting in cuttings, we use an auxin, we use IBA in talc or whatever. So auxin induces rooting. If we want to get shoots, if we want to get more shoots, we spray mild cytokinin on the growing branches. So cytokinin induces shooting. Gibberellins, it's an hormone known for bringing alongness, and same is its role in tissue culture. And ethylene is a senescence organ, so it induces maturation. So their roles remains almost similar in case of tissue culture. The most commonly used auxins are indole 3-acetic acid, that's IAA, naphthalene acetic acid, that is NAA, naphthoxy acetic acid, NOA, and 2,4-D, that is dichlorophenyl acetic acid. The response of these auxins varies. For example, we use IAA, NAA, and IBA largely for rooting purposes, but in many species, IBA is used for introducing callus. 2,4-D is the most potent agent for inducing callusing, and thus is used largely for somatic embryogenic purposes. Auxins, they cannot be dissolved in water, and we require either a weak alkali or a weak acid to dissolve them. So how we make the stock solution of auxin is you weigh the requisite amount, dissolve it in a weak alkali or acid, and then make the final volume. So by this you are not changing the media, and at the same time you are dissolving it fully. Cytokinins, they are largely responsible for cell division and differentiation of tissues from the growing callus. They are known to release axillary buds from apical dominance and thus help in shoot proliferation. The most commonly used cytokinin is BAP, that is benzyl aminopurine, 2IP, that is isopentenyl adenine, kinetin, thiodiazinon, and zeatin. BAP is the most frequently used cytokinin because it's the cheapest cytokinin and is most responsive in case of tissue culture. Zeatin, it has a limited role. It's the only natural occurring cytokinin, but it has limited role because it's very expensive and thus we only use it for research purposes. Like auxin, cytokinins also can't be dissolved in water, and we dissolve them either in a weak acid or alkali. So either HCl or NaOH is used for this purpose. I'll also like to add that some of these cytokinins, such as 2IP, they are heat labile, and thus we don't add to the media prior to sterilization. We add it to the molten media after sterilization through millipore filtration, and then pour the media in sterile vials thereafter. In some plant species, because of the continuous presence of cytokinin in the media, the shoots remain stunted and we cannot root them directly. So over there we introduce additional step of elongation, and in that media gibberellins are added. While range of gibberellins are available, more than 20 are known. GA3 is the most commonly used gibberellin in tissue culture media. Ethylene, which we call as senescence hormone. So this creates a sort of stress. The effect of ethylene in cultures is not clearly defined, and we do have examples which are both promotory as well as inhibitory. This usually is not a problem because when we put culture vials, there is a mechanism of air exchange. You will notice that in most plant tissue culture laboratories, cotton plugs are used. So cotton plugs in muslin, they permit air exchange, but then they don't permit any exchange or entry of microbes into the vial. Thus, usually in culture vials, ethylene deposition is not there and it is released as it is produced. There are also growth retardants which are known, for example, peclobutazole, which is used for specific purposes such as reducing hyperhydricity of leaves. And there are few others known such as encimidol favoring shoot formation in gladiolus and so on. But those are very specific, and of course it only tells you that when you are defining a media, it is not only the chemicals which are well established in tissue culture media, but you have to look beyond it. You have to study it. What is it that hyperhydricity, if it is caused, then what kind of chemicals can play a role, and so on. So tissue cultures can be raised in liquid as well as semi-solid. We avoid using liquid media largely because you have to maintain them on a shaker, as in static conditions the explant gets submerged and die due to lack of oxygen. In semi-solid media, the choice of the gelling agent is that it should be neutral. It should not add to any nutrients, or it should not avoid flow of any nutrients from it. And agar is the most commonly used gelling agent that is extracted from the red alga, Gelidium amensi. The unique property of agar is that it is liquid when hot at about 60 degrees centigrade and solidifies at 40 degrees centigrade. It forms a semi-solid gel that ensures that the plant tissue can be inserted properly and the nutrients can be absorbed by the explant from the lower cut end. It also remains solid on comparatively wide range of pH, anywhere between 5 to 6. I'll also like to mention that this alga can be grown readily. It is not expensive. Extraction process is not expensive. And we get different grades. One is highly purified grade, which is used for protoplast culture and so on, which is expensive. But the one which is used in most tissue culture, including micropropagation, is the food grade agar. Just to draw a parallel, what you eat at home as jelly is nothing but agar. So it is safe to eat, it doesn't add anything, and it is readily available. The most purified form of agar is agarose, but the purification is a lengthy, tedious process that adds to the cost. So again, the purified agar has limited application in case of protoplast and culture, as well as some of the genetic engineering experiments and so on. The third kind of gelling agent is gellrite. It is produced from a bacterium, Pseudomonas. It's easy to use, as unlike agar, it does not require any heating and can be prepared in cold water. The amount used again are comparatively lower, much lower. It's 0.1 to 0.2%, and it sets as a clear gel which assists in detecting any pathogen. So whenever you are testing your plant for endogenous bacteria or fungus, you put it on agar gel and immediately you can see a round thing appearing below. Agar is not transparent, it is translucent. So it's very difficult to detect any pathogen over here, which is far more simpler. But again, it's comparatively expensive, and more than that, it induces hyperhydricity in the leaves. Hyperhydricity is that the leaves, they become little brittle, they give glossy appearance, and they appear as if they are water soaked. So the hypohydric plants, the plants with hypohydric leaves, they fail to survive outside. So therefore, you don't require such plants, and therefore you don't use gellrite. It's also that combination of gellrite and agar has been successfully used in many tissue cultures. So it is combinations, different concentrations which have been used in tissue culture. Tissue culture can be regarded as an art, as there is no single approach to select a suitable media. So what do we do normally is that first we try with three kind of media: low salt, medium salt, and high salt. And we call it a checkerboard experiment. So you try a range of cytokinin and auxin combination. The most commonly used cytokinin here is BAP and auxin is Na. So you get a broad response, that is where a culture is responding, and then you fine tune it around it. Then you play around that, okay, it performed well in so and so auxin, approximately this concentration. Can I replace BAP with kinetin? Can I replace half of BAP with kinetin, and similarly Na with that? And what is happening on different salt media? If it's performing well on low salt media, it may not be that it requires low salt, maybe it requires a concentration of a particular salt to be reduced. So again, it is that you reduce macro concentration to 1/4, half, and then the individual salt, like you reduce total nitrogen by 50% or total magnesium by 50% and so on. And once you get some idea on it, then you move towards what other adjuvants will improve growth. And over there, you then play around with the various growth adjuvants which are known as well as the unknown ones. So this way you are standardizing what are the requirements of the major salts, minor salts, growth regulators, and if any unknown chemicals is also required. So in this module, you have learnt about media, its constituents, how those are prepared, and how we decide on what is required by the plant. Tissue culture media development, it's an art, and you have to work a lot to decide on what media is suitable to get a particular morphogenic response. But I must tell you that while you are developing media, you will come across with unexpected results. You will enjoy developing it. You will enjoy seeing how different kinds of responses are achieved on different kind of media. So happy learning. Thank you.